Hello, my name is AJ Stallsmith, and my presentation is on protein biosynthesis, aka protein synthesis. Today, we'll be discussing a few different topics about protein synthesis, starting with an introduction leading into historical developments, then into expert roles, positive and negative impacts, and finally ending with the conclusion. To introduce you to protein synthesis, I will be discussing the three main topics that make up protein synthesis, those being DNA, RNA, and polypeptide bonds. DNA is a molecule that carries genetic instructions for the DNA is a molecule that carries genetic instructions for the development, function, growth, and reproduction of all living organisms. It is a long, double-stranded helix composed of 4 nucleotide bases. The sequence of these nucleotide bases determine the genetic code and the characteristics of an organism. DNA is found in the nucleus of cells and in some organelles, such as mitochondria and chloroplasts. It is responsible for the transfer of hereditary information from one generation to the next. Secondly, we have mRNA. This is a type of RNA that plays a crucial role in the process of protein synthesis. It carries genetic information from DNA, which is transcribed into RNA to the ribosome, which is the site of protein synthesis. mRNA is produced during transcription, where a specific sequence of DNA is used as a template to synthesize a complementary mRNA molecule. The mRNA molecule then carries the genetic information to the ribosome, which uses information to synthesize a specific protein. The sequence of nucleotide bases determine the sequence of amino acids that make up the protein. Therefore, mRNA acts as a messenger, carrying genetic information from DNA to the ribosome, where it is translated into a functional protein. The next concept being a polypeptide chain. This is a long chain of amino acids that are joined together by amino acids. Amino acids are the building blocks of proteins, and a polypeptide chain is a precursor to a protein. During protein synthesis, amino acids are joined together in a specific sequence dictated by the mRNA molecule to form the polypeptide chain. Once the polypeptide chain is complete, it will often undergo folding and post-translational modification to form a functional protein. The sequence of amino acids in a polypeptide chain determines the shape, structure, and function of the resulting protein. Therefore, the sequence of nucleotide bases in the mRNA molecule, which determines the sequence of amino acids in the polypeptide chain, is critical for the formation of proteins. Now that we know the three main concepts, we can dive into this two-step process that is made up of translation and translocation. Translation is the process by which the information is encoded into an information sequence, which is used to synthesize a protein. It occurs on ribosome, which are complexes made up of RNA and proteins. The ribosome reads sequence in the groups of 3 nucleotides known as codons and matches each codon to the corresponding amino acids that it encodes. The amino acids are then linked together to form a polypeptide chain, which will eventually fold into a functional protein. Nextly, we have translation. Translocation, on the other hand, refers to the movement of proteins or other molecules from one location to another within a cell. In the context of protein synthesis, translocation refers specifically to the movement of ribosome along the mRNA molecule as the polypeptide chain is being synthesized. During translocation, the ribosome moves one codon at a time along the mRNA, while the newly synthesized polypeptide chain is transferred to the tRNA to the A site of the ribosome to the tRNA of the P site. This process continues until a stop codon is reached, at which point the ribosome disassembles and the newly synthesized protein is released. We got this picture from Jay Kapperman, and this was from Pinterest. Now, we will look at the historical developments. Here, we have a timeline that shows you some of the major discoveries, starting from 1902 and ending in 2000. Although all of these discoveries are important, a few of them really stand out. Are in 1953, when Linus Pauling discovered the formation of the double helix, as well as in 1961, when chemists Nuremberg and Mattei discovered the use of cell-free protein synthesis research. This picture here was from G. Towards Data Science. Historical developments, present. Nuremberg had paved the way to what we know as our modern day uses, these being muscle growth, diet, utilization of protein synthesis, and protein distribution. Some of our biggest takeaways would be understanding how to improve our overall strength and performance. We have learned recently about how protein synthesis is directly linked to performance within the human body. Historical developments, future. Scientists are just starting to break the barrier on what we call cell-free protein synthesis research. There is no doubt that creative application will go far beyond producing proteins and into fields like synthetic biology and medical diagnostics. Synthetic biology is an interdisciplinary field that involves the design, construction, and manipulation of biological systems for various applications. Some of the present-day uses of synthetic biology include biofuels, agriculture, medical applications, and industrial applications. Protein biosynthesis also plays a crucial role in modern medicine and has many medical applications. These include production of therapeutic proteins. Protein biosynthesis is used to produce therapeutic proteins such as insulin, growth hormones, and antibodies, which are used to treat a variety of diseases, including diabetes, cancer, and autoimmune disorders. Another one being gene therapy. Gene therapy is a technique that involves the delivery of genes into a patient's cells to treat or prevent disease. Protein biosynthesis is used to produce the therapeutic proteins encoded by the delivery genes. The last two being tissue engineering and drug discovery. Overall, the medical applications of protein biosynthesis are wide-ranging and have the potential to revolutionize the way we treat and prevent diseases. As a scientist, we look into the future for protein synthesis research, and we just keep seeing endless possibilities. Expert roles. Then, these research no field has occupied more investigators in biological science than in protein synthesis research. The two well-known chemists of this time are Nuremberg and Mattei, two names that won't be forgotten in protein synthesis research. Another name that won't be forgotten is a chemist who goes by the name of George Palade, who discovered that ribosomes perform protein synthesis in cells. He was awarded a Nobel Prize in 1974 for his accomplishments on this study. This picture here of George Palade was from The Famous People, and the picture of Nuremberg and Mattei are from Eve, Portugal, from the U.S. National Library of Medicine. Positive and negative impacts. Protein synthesis influences our lives every day. As an athlete, I am worried about maintaining a certain protein intake. This is to make sure I am allowing my body to perform and be as strong as possible. Another huge positive would be that there are jobs based on how good your muscles look. Jobs like bodybuilding and being a fitness trainer. Unfortunately, it's not all rainbows and sunshines. There is a negative side of protein synthesis. Recently, scientists have introduced perfect proteins. Anytime you genetically modify a natural process, it can lead to trouble. This is worrisome because protein synthesis errors can lead to toxic polypeptide chains. In some cases, protein synthesis mutations can cause genetic disorders such as sickle cell anemia, Huntington's disease, or cystic fibrosis. These disorders are caused by specific mutations that affect the function of important proteins in the body and have a range of symptoms and complications, including chronic pain, organ damage, and decreased lifespan. Conclusion. The problem is that protein synthesis is connected to every part of biology. This means that there is almost no limit to the amount of research that is able to be done. At some point, it has become too much, and scientists have had to decide what they were going to publish and what they weren't. Although there is much to learn from studies on protein synthesis research, it has slowed down because there is less of a need as there once was. Why is protein biosynthesis important? Protein biosynthesis is a fundamental biological process that is essential for life. Here are some reasons why protein biosynthesis is important. First being protein function. Proteins are essential molecules that perform a wide variety of functions in the body, including catalyzing biological reactions, as well as providing structural support to cells and tissues. Another function is gene expression. This is where protein biosynthesis is the process by which the genetic information encoded in DNA is used to produce proteins. As such, protein biosynthesis is critical for protein gene expression. This is the process by which genes are turned on and off to produce different proteins. As well, cellular processes is protein biosynthesis is involved in many cellular processes, including cell growth, differentiation, and division. Without protein biosynthesis, cells would not be able to grow and divide, and tissues and organs would not be able to develop and function properly. And finally, disease. Many diseases, including genetic disorders and cancers, are caused by defects in protein biosynthesis. Understanding the mechanisms of protein biosynthesis can help to identify the underlying causes of these diseases and develop new treatments. Overall, protein biosynthesis is a fundamental process that is essential for life and plays a critical role